Okay, today we're going to talk about fermentation, which is an anaerobic pathway. You can see listed here, meaning oxygen is not available. In fact, in most organisms, this is actually a backup mechanism when oxygen is not available. It is not preferred. The cell would actually prefer to go through cellular respiration, meaning to go through glycolysis, Krebs cycle, and electron transport chain to make the maximum amount of ATP, because if you go through fermentation, the ATP yield is only 2. Okay, so you only get 2 ATP from fermentation. All right, so let's look at a few things, see if I can get my pen up and running. Let's see, what am I going to put on here? So 2 ATP is the yield from fermentation. Where does it occur? It occurs in the cytosol, meaning that it's going to occur in the same exact place as glycolysis. Okay, so same place as glycolysis, meaning that it's not going to go into the mitochondria. So it is anaerobic, once again, happens in the cytosol. It's going to produce 2 ATP, and more importantly, though, the purpose, look right here by the purpose, it has to do with these NAD pluses. That's the real purpose. See, in glycolysis, if you remember correctly, glucose is oxidized to 2 pyruvic acids, or pyruvate. Okay, so we are going to change glycolysis, break it down, and we get 2 pyruvic acids. At the same time, here's what we got in glycolysis. 2 NAD pluses receive the electrons from that broken glucose and were reduced to 2 NADHs. Okay, and remember, these 2 NADHs, they headed over to the ETC. Okay, so they went over to the electron transport chain. Well, the problem is, if we don't have a way to recycle those and get back to NAD pluses, then the cell will actually deplete all of its available NAD pluses. So what we have to do is have a backup mechanism. In this case, when there's not enough oxygen available and the pyruvic acids do not go into the mitochondria and they don't go through the oxidative part of respiration, there has to be a backup mechanism in order to continue recycling NAD pluses. So in this bullet it says, in oxidative respiration, meaning Krebs cycle and ETC, NADH is usually oxidized back into NAD+. Okay, so it's going to be converted back after it drops off the electrons at the ETC. This goes back to being NAD+. So that normally happens over at the ETC. Okay, right here though it says, if oxygen is not available and these original pyruvates do not go into the mitochondria, then we're not going to be doing this. We're not going to be recycling these NAD pluses. So the cell must have another mechanism in order to regenerate that NAD plus, or glycolysis simply cannot occur. Okay, so in fermentation, here's the backup that we're talking about. This whole thing is about fermentation. What will happen is the pyruvate, or pyruvic acid, stays in the cytosol, goes through some changes, can be reduced, which will oxidize our NADH to NAD+. So it allows our cell to still continue to go through glycolysis by taking the pyruvate, which was formed in glycolysis, and continue changing it into something else. And so this allows glycolysis to continue and allows us to still continue to produce 2 ATP. So it is a backup mechanism. Okay, now, there are 2 types of fermentation. Okay, the first type you see listed down here, this is alcoholic fermentation. Okay, the one on the next screen is lactic acid fermentation. So there's alcoholic and lactic acid. And in this first slide, I just kind of want to spotlight here for you. Get rid of this a little bit. Okay, in this first slide, this is alcoholic fermentation. Once again, this is happening in the cytosol. You might want to note that on your notes. Okay, out in the cytosol. And we're going to start off with our pyruvate. Here's what we're starting off with right here. Okay, now where did that come from? Remember, that came from glycolysis. You can see right here, glucose was broken down in glycolysis to give us pyruvate, or pyruvic acid, which is a 3-carbon compound. See, 1, 2, 3-carbon compound. Okay, now when we did that, let's go back and review real quick. We generated 2 ATP. Remember, we had the energy investment stage and the energy yield stage, and so we ended up producing 2 ATP from that. Okay? In addition, we had NAD pluses that received the electrons from us breaking glucose and were converted into 2 NADHs. So in glycolysis, we get this right here. We get 2 ATP, we get glucose split into pyruvate, and we get NADHs that will go over to the electron transport chain. Okay? Now, if oxygen was not available, okay, no oxygen over at the electron transport chain, and remember the job of oxygen at the ETC is to catch the electrons at the very end. If there's no oxygen, this pyruvate stays out here in the cytosol and does not go into the mitochondria. In that case, there won't be any more NAD pluses after a while because the cell will always go through glycolysis, and as we do this, we'll use all the NAD pluses. So this backup mechanism, alcoholic fermentation, is where our cell takes this pyruvate, okay, breaks it, we do some changing to it, we break off a CH in this case, okay, one of the carbon atoms. Okay, it's going to leave as a carbon dioxide molecule, but we're left with a 2-carbon compound known as acetaldehyde. Okay, that then is less toxic than the pyruvic acid was because too much pyruvic acid in the cell can actually be toxic to the cell. So now we have a less toxic compound, but more importantly, that can be changed by adding hydrogens onto it, okay? So we can add some little electrons and hydrogens onto it to produce ethanol, which is ethyl alcohol. So by doing that, we can cleave this hydrogen off of NADH. This goes back to NAD+. The cell is happy because glycolysis can continue due to the fact that we have this NAD+ again. And now we have something called ethyl alcohol. Now, just so you know, we do not do this. This is not in humans. Okay, alcoholic fermentation happens in bacteria, and also in yeast cells, which yeast are funguses. But this occurs in yeast and bacteria, and it is important to us because the yeast that utilize this is one of the main ways that we make bread. Okay, and so when we make bread, you know, the rising of the bread while it's cooking, the CO2 over here, one of the waste products, is what makes bread rise. Okay, the ethanol that's in the bread, when the bread bakes, it actually dissipates out. But also in the cheese and dairy industry, they use this type of they use lactic acid, just kidding. In the brewing industry, in the brewing like beverages, beer, wine, champagne, etc., this is also the process that's used in that, and so the carbon dioxide is the bubbly that you see, and then the alcohol is formed as a byproduct. Okay, so that's alcoholic fermentation. Now, in the cheese industry, that's the next slide, which is lactic acid fermentation. Okay, this is utilized by some bacteria in producing cheese, so this is important to us for that reason as well, but this is also one that occurs in humans, okay, because we have to have one in us, and this is the human one. It's called lactic acid fermentation. And once again, glycolysis is still going to occur. You still get the same steps from here to here. We still get our ATP, we get our glucose broken down, and we get our NAD+ reduced to NADH. Okay, but in this one, the product this time is going to be lactate or lactic acid. Okay, so a slight change. So in lactic acid fermentation, there is no production of carbon dioxide. The pyruvate is simply changed as it receives the electrons from NADH. So what we like to say is the pyruvate becomes reduced because electrons are coming to it from NADH. And once that happens and we form this lactate or lactic acid, then we have available NAD pluses, which can continue the process of glycolysis. So this occurs, like I said, in human muscle cells. In us, when this would happen is when we are exercising vigorously. So if you're out there, let's say, doing wall sits or doing crunches or doing something for an excessive amount of time, our oxygen supplies are depleted, and as oxygen decreases, then the pyruvic acid cannot enter into the mitochondria and go through oxidative respiration, and so it stays out in the cytosol and goes through this alternative pathway. Okay, so once again, this one can be utilized by yeast, bacteria, and humans, okay, and it's lactic acid fermentation. The benefit for us is it continues glycolysis in our muscle cells so that we can continue to break down sugars. We also get the dairy industry is producing cheese and yogurt for us, which we do utilize. So that's a benefit in that regard. Okay, going back to this one, benefit is production of alcoholic beverages, production of breads, things like that. But once again, both these pathways occur in the absence of oxygen, but the benefit is to keep the recycling of NAD+ so we can keep glycolysis moving. Okay, and doing that, our pyruvate will be reduced by accepting electrons from NADH and becoming a different product. So in this case, it's ethanol, and in the case of this one, it's lactate. All right. Okay, now let's talk briefly. We're going to jump back for a minute and talk about photorespiration, and this will conclude, after I do this, this will conclude all the photosynthesis information, and we will be 100% done. Woohoo! Okay, so this slide talks about an evolutionary kind of relic known as photorespiration. So let's look at the diagram for a second. In most plants, initial fixation of carbon, right here, the fixation of carbon, occurs via rubisco, the magical enzyme, the Calvin cycle enzyme that adds carbon dioxide to rubus. Okay, such plants, the plants that do this that we talked about, are called C3 plants because the first organic product of carbon fixation is a 3-carbon compound, that 3-phosphoglycerate. Okay, rice, wheat, and soybeans are types of C3 plants that are important in agriculture. That's how we get a lot of our food. When their stomata partially close, like on a hot, dry day, these C3 plants produce a lot less sugar because of the declining levels of CO2 in the leaf. So it kind of starves the Calvin cycle. In addition, rubisco can bind oxygen in place of carbon dioxide. So as carbon dioxide becomes scarce during a hot, dry day, okay, so remember this is like a declining level of moisture in the air. So as the carbon dioxide decreases within the airspace of the leaf, rubisco can actually add oxygen to the Calvin cycle instead of adding carbon dioxide. This product splits and the 2-carbon compound leaves the chloroplast. So instead of getting a 3-carbon compound, we get a 2-carbon compound. Okay, now we're going to talk about how, like, actually remember the organelles called the peroxisomes in the mitochondria? They'll be there inside the cell and they will help rearrange and split this 2-carbon compound, which will inevitably result in the release of carbon dioxide. Okay, so you can see over here on this side, it'll take in the oxygen, it'll rearrange it to a 2-carbon compound. Some of the carbon dioxide is released as the peroxisomes and mitochondria rearrange that. Okay, and this process that does this is called photorespiration. Now, it occurs in the light, which is the photo part, and it consumes oxygen while producing carbon dioxide, which is where the respiration part of the word comes in. Okay, so it's not exactly like cellular respiration. In fact, it's a little bit of an opposite because there is no generation of ATP in this. In fact, it's consuming ATP. It takes energy for them to do this. And unlike photosynthesis, it produces no sugar. So your sugar amount decreases as far as the plant is concerned. So it decreases that photosynthetic output by siphoning organic material from the Calvin cycle and releasing the carbon dioxide that would have normally have been fixed. Okay, so this is a evolutionary process that happens in times of, like I said, like really hot, dry days when the stomata on the leaf would close, which would decrease the amount of carbon dioxide coming into the leaves. But for them to keep on with the cycle, they would go through this photorespiration. And, one more time, just to show you in the chart, they would take oxygen as opposed to carbon dioxide, which would be high present and would be there in the leaves, and they would use that to assemble a 2-carbon compound, which would then be broken down by peroxisomes and mitochondria, using a little bit of energy in releasing some carbon dioxide. So it decreases this energy out. Decreases the carbon yield in this, so we're making less and less sugar. Now, you might be wondering, why would plants do this? They have discovered that in most cases, photorespiration is a protective role in plants because plants that are impaired in their ability to carry out photorespiration are more susceptible to damage by excess light. It kind of shows that this process, photorespiration, acts to neutralize the otherwise damaging products of the light reactions, which would build up when a low carbon dioxide concentration limits the progress of the Calvin cycle. Okay, now in this slide, we're going to talk about something that's really cool. It's an evolutionary adaptation. In some plant species, alternate modes of carbon fixation have evolved that minimize the need for photorespiration and optimize the Calvin cycle, even during hot, arid climates, The 2 most important of these photosynthetic adaptations are, you can see one here, it's called C4 plants, okay, C4 plants, and then also CAM metabolism, which stands for crassulation acid metabolism. Crassulation, that's kind of weird. Okay, so C4 plants over here are so named because they preface the Calvin cycle with an alternative mode of carbon fixation that forms a 4-carbon compound as its product. There are actually several thousand plant species in many different plant families that use the C4 pathway. Most important, as you can see, one listed here is sugarcane, but also corn. And like I said, this is an alternative pathway to have to not use photorespiration, but still be able to produce a product. So one of the things that you can see in the diagram is this mesophyll cell, bundle sheath cells. They have a really unique leaf anatomy that is correlated with the use of these special kind of cells. Bundle sheath cells down here at the bottom are arranged into tightly packed sheaths around the veins of the leaf, and the Calvin cycle is confined to the chloroplasts that are in these bundle sheath cells. However, the Calvin cycle is preceded, you can see right before, by the incorporation of carbon dioxide into organic compounds in the mesophyll cells. So they're still able to produce a product in these hot, dry, arid climates and still be very successful despite the fact of not having to increase photorespiration. Okay, on this side over here, the cam plants, you can see pineapple in the picture because that is a type of cam plant. Other cam plants besides pineapples are lots of our succulents, and we actually have, you guys might have some in your own house or your own yard, but lots of cacti, pineapples, different types of succulents. They have this second evolutionary adaptation, okay, these cam plants. What they do is kind of cool. In any time that there's an arid condition, okay, so it's hot and dry, these plants do something different. They actually open up their stomata, okay, the openings in the underside of the leaf that allow for gas exchange. They open up their stomata during the night and then close them during the day, just the reverse of how other plants behave. Closing the stomata during the day helps desert plants conserve water, but it also prevents carbon dioxide from entering the leaves. During the night, when the stomata are open, these plants can actually take up CO2, incorporate that carbon dioxide into many of their organic acids. So they're still going to go through the light reactions during the day, do the normal thing with the light reactions, exciting the photosystems, and getting what they need, but then at nighttime is when they'll open up those stomata, and fix the carbon into their sugars at night. And then just to finish it off, just to show you down here at the bottom, I didn't know if you saw the A, how it was labeled under C4 plants, and spatial separation of steps, meaning that during this process it's in two different types of cells to go through the incorporation of the carbon dioxide and then the production of the sugar, whereas in cam plants, the temporal separation means day and night, 2 times of the day in order to get the carbon dioxide and then use it for the sugar. So that concludes our whole entire unit, so we are done.